Good evening, all. This is our SYJC biology lecture. We are in chapter number 3. Name of the chapter is Inheritance and Variation. We are in part 4 of the chapter. I am Professor Prasad Tavankar. We will be discussing something related to what is said as chromosomal basis of inheritance. Now, I told you this chapter has got two basic divisions like basis of inheritance and chromosomal basis of inheritance. So we have to understand what is meant now by chromosomal basis of inheritance. Actually, we have started this last time and we did chromosomal theory, which was given by Sutton and Boveri. So two scientists gave this particular theory, and it says that all the cells have got nucleus, of course, except viruses. Viruses don't have got nucleus in their body. That's what viruses stand different. Then nucleus consists of genetic material. Genetic material is in the form of chromosomes arranged in a proper manner. At the time of gamete formation, the chromosomes segregate, homologous chromosomes segregate, and at the time of fertilization, they reunite. So these are some of the postulates of chromosomal theory. So that's what we did till last time. Now we have to go ahead with this particular thing. We have to understand now chromosome morphology, chromosome structure, number of chromosomes, etc., etc., etc. We have to understand now, and we'll carry our story further. So as far as chromosomes are concerned, you must know chromosomes are present in nucleus. As per this picture, as it is displayed over here, you could see that there is a nucleus present. Nucleus consists of chromosomes present in it. Chromosomes are very densely packed, means there is a DNA present as well as proteins present. DNA and proteins are available in the ratio around, we can say, 33 to 67%. So 1/3 is to 2/3 is DNA and protein. Uh, that will understand once again in another chapter why proteins are required. Proteins are required for binding DNA tightly. So that's how DNA will be bounded with the help of proteins very tightly, very compactly, and then ultimately it is DNA. DNA has got some fragments on it. Those fragments we call them as genes. Genetic basis we have understood. Now chromosomal basis. We need to understand the morphology of chromosome. Chromosomes were discovered by Wilder. Rather, we can say they were named by Wilder. Why they were named by Wilder like chromosomes? Because there are two words. Chroma means color, you must be knowing, and soma means body. So chromosomes are the colored bodies. When scientists were trying to understand what are chromosomes, they tried to stain the cells. अभी सेल को स्टेन करना शायद आपको नहीं पता होगा because you have not been to biology laboratory till now. Okay, 11वीं में 11th standard में आपको biology laboratory में जाने को नहीं मिला, but hope you will be able to reach 12th standard biology laboratory directly, where you will be using different colors to stain the cells. Now why these stains are required? Stains का जरूरत क्या होता है? अलग अलग कलरिंग मटेरियल है. These are actually different chemicals. These chemicals would be accepted by different parts of the cell and they get different colored. So different parts of the cells get colored differently, and that will make us to understand that these cells are having these parts. Or sometimes different tissues get colored differently. So depending upon the color that is accepted by a particular tissue, we can say yes, this is particular tissue is. तो कलर करने से फायदा होता है कि हम लोग आइडेंटिफाई कर सकते हैं. सेम थिंग वाज ट्रू अबाउट दिस पर्टिकुलर थिंग. So when chromosomes, when the thread-like structures were stained, they accepted color, and that's how they were called as chroma means color and soma means body. So colored bodies, that's what was the meaning. Okay. Chromosomes are invisible. They are available in the form of a long, thin kind of threads spreaded in the nucleus in the ordinary cell. But whenever cell enters in a cell division cycle, just prior to cell division, in the prophase stage, the chromosomes start condensing. They start becoming short. They start becoming thick, and now they are visible. So earlier they were invisible. Now they are visible. Now these chromosomes, they are capable of self-duplication, and that's how they are capable of maintaining heredity as well as variation both. Now, why chromosomes are capable of self-duplicating? Concept is very simple. Actually speaking, it is the DNA present in it. This DNA is capable of undergoing replication process. DNA replication का मतलब क्या होता है? There is a process of formation of two DNA molecules from one single DNA molecule. That's what is called as DNA replication. And so because of this, entire chromosome also will get doubled. So that is what the basic is. And now the chromosomes are equally distributed. Morphologically and genetically similar chromosomes are known as homologous chromosomes. We have given this concept already earlier. At that particular time, we said the chromosomes, two or more chromosomes, rather two chromosomes generally, which are having same morphology. Morphology same है मतलब क्या? They are having same length, same position of centromere, etc. And they are having same genes at same loci. That's why they're genetically also similar. Same genes तो नहीं हो सकते, बहुत बार similar genes होते हैं. Means genes for alternative characters or allelic genes. When are present, the chromosomes are said to be homologous chromosome types. So that is how available. The number of chromosomes is definite in each cell. That is also we have said earlier in the chromosomal theory. We have said this particular point. The number of chromosomes is fixed in a particular species. If the number of chromosomes changes, the species will be different. Now, so far as the number is concerned, we have to understand certain terms regarding the number of chromosomes that are present. First is euploidy. U का मतलब होता है advance. Ploidy is set of chromosomes. If a cell has a normal set of chromosome or one particular entire set of chromosome, then it will be called as euploidy. ठीक है? So this euploidy may be duplicated either once or several times. So one particular thing you have to remember, that is euploidy. Euploidy के बाद आएगा अपने को that is haploidy and diploidy. Now haploidy, diploidy you might be aware of. Haploid is one single set of chromosome, so that is called as haploidy. Single set of chromosomes is indicated by N. So if you are asked what is haploid, you should not say N, you should say it is one set of chromosomes. Okay. This is common state for gametes. So generally gametes are haploid, of course, for those organisms which are undergoing sexual reproduction. For organisms undergoing asexual reproduction, haploidy, diploidy doesn't matter, because asexual reproduction, generally there is mitosis available. So mitotically cells will keep on dividing, number of chromosomes remains same. But for sexual reproduction, haploidy diploidy matters because gametes are haploid and normal cells, somatic cells, our bodily cells are diploid. So diploid का मतलब होता है two sets of chromosomes indicated as 2n, and it is common for somatic cells or normal set of body or normal cells of body. There is a condition called as triploidy. So 3n, three sets of chromosomes are present. If you say tetraploidy, it will be four sets of chromosomes. Likewise, pentaploidy, hexaploidy, etc. You can keep on saying. Or polyploidy, many chromosomes will be there. Many sets of chromosomes will be there. Now let us come to human. Human cell, if you take into consideration any body cell of our body other than gametes, अपने बॉडी का कोई भी सेल उठा लो, even germinal cells are those which are forming gametes. So every cell of our body has got 2n number of chromosomes, means they are diploid in nature. So there are two sets of chromosomes. These two sets equal to 46 chromosomes or 23 pairs of chromosomes. अभी ये chromosomes देने का रिवाज अलग अलग से है, मतलब एक ही नंबर हम लोग बताएंगे बट अलग अलग ढंग से उसको बताएंगे. We may say 46 chromosomes or we will say 23 pairs. Each chromosome is available in double dose. Double dose का मतलब क्या होता है? हर एक क्रोमोसोम अपने कॉपी के साथ आता है. कॉपी मतलब अपने जैसे दूसरे क्रोमोसोम के साथ आता है. That's what is actually called as homologous chromosome pairs. I have told you already earlier, one chromosome of homologous chromosome comes from mother, other comes from father. So when we say homologous chromosome pair, you have to be sure that one chromosome of this is belonging to father or paternal family and other chromosome is belonging to mother or maternal family. Maternal family मतलब मम्मी का family, paternal family मतलब पापा का family. So इस तरह से अपने को जोड़ी में chromosomes होते हैं. Now amongst this there are 22 pairs of chromosomes that are called as autosomes. Okay, autosomes का मतलब क्या होता है? These chromosomes are not involved in sex determination or involved in sex formation. Okay, that's why they are called as somatic chromosomes. Soma मतलब body बनाने में मदद करेंगे. That's why somatic chromosomes or autosomes or non-sex chromosomes. But there is another pair or single pair of chromosomes is present in our body, that is known as sex chromosomes or godosomes और heterosomes. वो बाद में आएगा क्यों अलग अलग नाम उनको दिए गए हैं और क्या कहलाते हैं ये. Average size of chromosome, when we are studying morphology of chromosome, we should remember the size of chromosome. It will vary from species to species. It is not constant. The size of chromosomes is different. But in case of metaphase state, the chromosomes will appear around 0.1 to 0.33, sorry, 0.1 to 33 microns in length and 0.2 to 2 microns in. Remember this, 0.1 to 33 microns. 1 micron, you must be knowing, 1 micron is 1,000th part of millimeter. So, kitna chota rahega, just try to believe or imagine. So, it is 0.1 to 33 microns in length and 0.2 to 2 microns in thickness. Then, as far as structure of chromosomes is concerned, chromosomes are having basic fundamental structure that we have to understand. As we have said, they are best visible under microscope during metaphase stage of cell division stage. Okay. At this particular stage, chromosomes are highly condensed. I told you about this. Chromosomes keep on condensing, means they become shortening, they become thickening, and now they become visible. Okay. Earlier, the chromosomes are long, thread-like, thin, and dispersed state. Now they become short, condensed, thick state. Typical chromosome will be having two chromatids present in it. Okay, they are generally called as sister chromatids. अभी sister chromatids मतलब feminine दिखाया है word यहाँ पे. So two chromatids are available as they look exactly alike. Just try to understand what I am saying. एक chromosome के दो chromatids होते हैं and they are exactly alike or exactly similar. Try to recall the things which we discussed while understanding cell division chapter in 11th standard. So exactly alike, similar chromatids are exactly similar. They don't have got any difference. Okay, and they are connected together or joined together with a small structure that is known as centromere or primary constriction. Centromere को ही कहा जाता है primary constriction. It consists of disc-like structures or plates present in it. Those disc-like plates are called as kinetochore. Now, kinetochore of each chromatid is different and they are held together with the help of the bonds present between them through the primary constriction. तो अंदर क्या होगा? Kinetochore होगा. These kinetochores are the points where the chromosomes are attached by spindle apparatus or spindle fibers. क्रोमोसोम को खींचना है ना, दो क्रोमेटिड्स को अलग अलग करना है. ड्यूरिंग सेल डिवीजन इट इज नेसेसरी दैट द क्रोमोसोम शुड बी separated from each other. At that particular time, if chromosome is connected to spindle fiber at any other point, it may be possible that the chromosome may break. मैंने क्या कहा समझ में आया? क्रोमोसोम को अगर स्पिंडल फाइबर अगर क्रोमोसोम को कहीं भी पकड़ लेता है अदर देन काइनेटोकोर, देन क्रोमोसोम्स आर वेरी फ्रेजाइल. चांस है कि क्रोमोसोम टूट सकता है. तो क्रोमोसोम को टूटने से बचाने के लिए it is very essential that it should be connected to a one single point, and that is nothing but kinetochore, or we can say even primary constriction, or we can say even centromere. Besides this primary constriction, the chromosomes might be having secondary constrictions or tertiary constrictions. Generally, they are called as secondary constrictions. They may be either one or two or maybe many in some cases, depends on the different chromosomes. These are called as secondary chromosomes or secondary chromatids or secondary constrictions. Remember it very well. Now, this is the general idea so far as chromosome is concerned. You could see over here, this single is called as one entire chromosome present in a mature cell. Mature cell का ये पूरा chromosome है. And you could see this point is a point where the two chromatids, now ये वाला एक जो part है उसको कहेंगे one chromatid, ये वाले को कहेंगे दूसरा chromatid. So each chromosome is composed of two chromatids when matured, and they are connected to each other at this particular point. This is called as primary constriction. Of course, there are many other constrictions possible, but this is primary constriction available. At the time of cell division, spindle fibers would get attached over here, that is to kinetochore, and then they will be separated. So each chromosome consists of two sister chromatids. Mind it very well. So that's an idea. At secondary constriction, now secondary constrictions क्या है? जैसे primary constriction है वैसे secondary constriction है. There may be one or two, we have said. So at secondary constriction 1, what will happen? The nucleolus becomes organized during interphase. That's why it is called as nucleolar organizer. Primary constriction को क्या, secondary constriction को क्या कहा जाता है? Nucleolar organizer. So what is nucleolus? Nucleolus is a subnuclear structure where each and every chromosome, secondary constriction of each and every chromosome will contribute, and this is a place where RNAs are formed, ribonucleic acid are formed. So that's an idea behind it. A satellite body or SAT body will be attached to the chromosome at secondary constriction. Okay, मतलब एक और constriction यहाँ पे रहेगा. So अपना primary constriction है, बाद में secondary constriction 1, secondary constriction 2. Secondary constriction 2 is generally present towards the tail end of chromosome, which will separate a small hanging body, we can say, so of chromosome, of course, and it will be connected to the chromosome, main chromosome part. This particular hanging body will be called as satellite chromosome. It is not a property of all chromosomes. So satellite chromosome is not found in all chromosomes. It is a property of few chromosomes only. So primary constriction essential for holding the two chromatids together. Secondary constriction 1 is essential for formation of nucleolus or nucleolar organizer, and constriction number 2, secondary constriction number 2 is essential for formation of satellite chromosome. Now each chromatid, ये chromatid है, हम लोग बता रहे हैं, one chromatid is in turn consisting of a long, unbreakable or unbranched, slender, highly coiled DNA present in it. Okay, हर एक क्रोमैटिड के अंदर डीएनए होगा. मैंने बताया पहले एक स्लाइड आपको दिखाया था, उसमें एक पिक्चर दिखाया था कि न्यूक्लियस है, न्यूक्लियस के अंदर आपको क्या दिख रहा है? क्रोमोसोम दिख रहा है. एंड इफ वी कीप ऑन अनवाइंडिंग क्रोमोसोम विल अल्टीमेटली रीच अप टू डीएनए. ठीक है, जिसको कहेंगे पैकेजिंग ऑफ डीएनए. डीएनए का पैकेजिंग कैसे होता है हम लोग बाद में सीखने वाले हैं. ओके, सो दिस हाईली कॉइल्ड डीएनए थ्रेड इज कंसिडर्ड एज क्रोमोनिमाटा. ठीक है, chromonemata, and many chromonemata will show chromomeres, मतलब parts of the chromosomes will be also seen on the chromosome. It extends through the length of chromatid, मतलब पूरे chromatid में एक जगह से लेके दूसरी जगह तक ये chromonemata रन करेगा, thread बनेगा. See, DNA undergoes coiling in a definite fashion. Okay, and then it keep on becoming larger and larger thread, means bigger and bigger thread. ये अभी बाद में हम लोग सीखेंगे तब ज़्यादा डिटेल में पता चलेगा. So at that particular time, once again, if you want, we will understand how the chromosome is formed. So that's how chromonemata will be there. Then chromatid will consist of double-stranded DNA molecule, which extends from one end to other end. That's what I told you just now. So this is how we can say generalized structure of chromosome. This is centromere. Then here we can say euchromatin and heterochromatin. Of course, these are the parts of chromosome. We have understood. Sorry, what is euchromatin? Euchromatin means advanced chromatid. This is loosely packed, and heterochromatin is tightly packed. Euchromatin is a part of chromosome which is undergoing what we can say expression of genes. Okay, so it is functional part, and heterochromatin is non-functional part of chromosome. Both will be seen. Heterochromatin will be appearing densely packed and dark colored structure, whereas euchromatin will appear loosely packed and light color structure. Okay, there is one more part available to the chromosome, that is known as telomere. Now telo is referring to tail or end of the chromosome, and so that is end of the chromosome or end part of the chromosome. It will be responsible for undergoing repetitive sequences of non-coding DNA. अभी ये आपको शायद नहीं समझेगा क्योंकि कोडिंग डीएनए नॉन कोडिंग डीएनए आपको वर्डिंग मालूम नहीं है. DNA has got nitrogenous bases, and these nitrogenous bases are forming genetic language and they code for a particular amino acid. But यहाँ पे ये जो पार्ट है टेलोमियर का पार्ट है उसमें नॉन कोडिंग डीएनए अवेलेबल है, मतलब उसमें से कोई प्रोटीन नहीं बनेगा। सिंपली अभी इतना ही याद रखो, टेलोमियर जो है, टेलोमियर के जो डीएनए है उसमें से कोई भी प्रोटीन नहीं बनेगा। बाकी के क्रोमोसोम में जो डीएनए है उसमें से प्रोटीन बन सकता है, लेकिन यहाँ से नहीं बनेगा। ठीक है, इतना ही इन शॉर्ट अपने को अभी याद रखना है। सो फार एज व्हेन वी आर सेइंग नॉन कोडिंग डीएनए इज अवेलेबल, इट विल बी रिस्पॉन्सिबल फॉर प्रोटेक्टिंग क्रोमोसोम फ्रॉम डैमेज। तो टेलोमियर का काम क्या है? दो काम है उसके। इट विल गिव प्रोटेक्शन टू द chromosome from damage, and secondly it will avoid sticking of chromosomes to each other. Chromosomes कभी भी एक दूसरे को चिपकेंगे नहीं, that is because of telomere is present, and chromosomes will be always protected, that is also because of telomere. Okay, interesting thing is telomere will keep on dividing and redividing along with the cell division, but the important thing is whenever cell division takes place, whenever chromosomes divide, DNA divides, the telomere undergoes division, and every time the cell divides it will keep on becoming shorter and shorter. A stage comes when the telomere will become very short, and when telomere becomes very short, it is a state where our cells cannot survive now, the cells will die. Okay, so cells के डेथ के लिए या सेल्स के एजिंग के लिए टेलोमियर इंपॉर्टेंट है। ओके, अगर टेलोमियर, मैंने क्या कहा समझ में आया? टेलोमियर will keep on dividing, but every cell division will make the length of telomere short. A stage will reach at which the telomere will be very short. Now it cannot divide further, and so cell division will stop. Cells का division बंद हो जाएगा, cell division नहीं होगा, and that's how the death of cell is. So whenever more number of cells are dying, it is aging, and that's how we end our life. ठीक है, सो देखो जो बनाने वाला है उसमें सब कुछ सिस्टमैटिक करके रख दिया है, कुछ भी प्रॉब्लम नहीं है यहाँ पे। फाइन, सो साइंटिस्ट आर वर्किंग ऑन दिस। What scientists are working on this? They are working on telomeres. You must be knowing organism that survives for the most life. सबसे ज्यादा जिंदा रहने वाला ऑर्गेनिज्म कौन सा है? You must be knowing it is cockroaches. Is it? No, they are, what are they? Turtles. Turtles survive for many days, around 250 to 300 years they survive. So what is special in them? टर्टल्स के क्रोमोसोम्स में क्या ऐसा स्पेशल है, टर्टल्स के टेलोमियर्स में ऐसा क्या स्पेशल है दैट मेक्स देयर लाइफ मोर? लाइफ स्पैन बहुत ज्यादा हो गया ना, अपना लाइफ स्पैन करीब करीब 80 साल हो गया, अभी 70 विल बी एवरेज। फाइन, सो इफ वी वांट टू इंक्रीज अवर लाइफ स्पैन, then we have to make a research on telomere, and we should take proper care that telomere will not decrease after every cell division. If it is possible, then our lifespan can be increased. ओके, ये कंसेप्ट होते हैं इस तरह से। तो इस तरह से हम लोग बता सकते हैं कि अपना लाइफ स्पैन बढ़ाने के लिए अपने को टेलोमियर के ऊपर काम करना पड़ेगा। आप लोग बोलोगे कितना लाइफ स्पैन की जरूरत क्या है, इतना क्या करने का है? इट इज एसेंशियल फॉर स्पेस एक्सपेडिशन। अगर हम लोग को बाहर जाके एक्स्ट्रा टेरेस्ट्रियल जैसे कि मार्स के ऊपर जाना है आदमी को, तो जाने के लिए 7 महीना लगेगा, आने के लिए 7 महीना लगेगा, विद द हेल्प ऑफ फास्टेस्ट व्हीकल दैट वी आर हैविंग, है ना। अभी आपने मंगल मिशन देखा रहेगा या कोई और अपना ऐसा अपने नासा का भी देखा रहेगा, अभी रोवर उतर गया है वहां पे, तो जाने के लिए 7 महीना लगा हुआ है सैटेलाइट को उधर पहुंचाने के लिए। ठीक है, सो इफ वी वांट टू ट्रैवल इन स्पेस, मोर लाइफ इज रिक्वायर्ड, ताकि आदमी जाएगा स्पेस में, कुछ काम करेगा और काम करने के बाद आके वो वापस रिपोर्ट करेगा, इसके लिए लाइफ स्पैन ज़्यादा चाहिए, एंड फॉर दैट मोर लाइफ स्पैन we should work on this. Fine. So this is a picture of chromosome indicating this area is telomere. This is entire chromosome. This is one single chromatid, or this is second chromatid. So both are called as sister chromatids. This part is telomere. इसको कहेंगे secondary constriction. Then this is primary constriction or centromere. And you could see here a disc-like structure that is called as kinetochore. This kinetochore is useful for, I told you, connecting spindle fibers to it. Okay, then you could see heterochromatin over here, and you could see even euchromatin somewhere if it is possible. Yes, this is euchromatin part and this is heterochromatin part. Okay, so loose part and condensed part, that's how we can take into consideration. These are different areas, functional and non-functional areas. Okay, so that's जनरल आइडिया. क्रोमोसोम का स्ट्रक्चर बनाने के लिए बोलेंगे तो आपके बुक का डायग्राम जो है वो आपको रिफर करना पड़ेगा, एंड यू टू मेक अ डायग्राम लाइक दैट. ये डायग्राम ड्रॉ करने के लिए 2 मार्क के लिए आएगा. क्रोमोसोम का मॉर्फोलॉजी के साथ क्वेश्चन पूछा गया तो 4 मार्क के लिए आएगा. सो यू हैव टू गिव ऑल द पार्ट्स ऑफ क्रोमोसोम्स एंड डिस्क्राइब इट इन अ वेरी वेल फैशन. Fine. So that's how we have to understand about the morphology and structure of chromosome. Now we have to understand the classification of chromosomes. Chromosomes are classified in a different way, and one basic of it is nothing but the position of centromere. So chromosomes can be divided, or chromosomes can be classified on the basis of their size first. Larger chromosomes, medium chromosomes, and small chromosomes. Then second comes the position of centromere. Depending upon the position of centromere, chromosomes are of 4 different types, namely metacentric, submetacentric, acrocentric, and telocentric. Try to observe it from right to left. Generally हम लोग left to right पढ़ते हैं, but इस तरह से याद रखोगे तो अच्छा लगेगा और याद रखने के लिए आसान हो जाएगा. This one is called as metacentric chromosome. Why metacentric chromosome? Try to observe the position of this centromere. Centromere is exactly at the center. Centromere is exactly at the center of the chromosome or chromatid, and that's why it is called as metacentric chromosome. Because of this, each chromatid will be having 2 arms formed. One is called as P arm, other is called as Q arm. Okay, so P arm and Q arm, when you take into consideration, these arms are equal over here. Why they are equal? Reason is very simple. क्या रीजन है? यहाँ पे P arm Q arm सेम होने का, सेम लेंथ का होने का रीजन क्या है? Because centromere is at the center. That's why chromosome is called as metacentric type of chromosome. Second one we are going to say is submetacentric chromosome. Centromere here is present nearer to the center, but not exactly at the center. यहाँ पे क्रोमोसोम का सेंट्रोमियर कहाँ पे है? सेंटर के पास है, बट जैसे इस तरह का एग्जैक्टली सेंटर में था, यहाँ पे एग्जैक्टली सेंटर में नहीं रहेगा. And that's why P arm and Q arm are not equal. P arm is shorter, Q arm is longer. Okay, so that's an idea behind it. Then third comes acrocentric chromosome. Acro means height or tail or end. So acrocentric chromosome is that in which the chromosome is present, centromere is present at the end of the chromosome or nearer to the chromosome, nearer to the end of chromosome, but not exactly at the chromosome. Okay, so in this case what is happening? Chromosome has centromere towards the end but not exactly at the end, and that's how P arm will become very short and Q arm will become very long. And last category is acrocentric chromosome, telocentric chromosome. Telocentric chromosome is that in which tail end, मतलब tail में end में ही रहेगा. So one arm has gone and other arm is available. So P arm is almost absent, Q arm is present. That's what is the characteristic feature. Okay, so 4 types of chromosomes depending upon position of centromere as metacentric, centromere is at the center exactly. Submetacentric chromosome, where chromosome has centromere nearer to the center but not exactly at the center. Then there is third acrocentric, centromere is present towards end but not exactly at the end. And fourth is telocentric, in which the centromere is exactly at the end. So this way we can say 4 types of chromosomes. Now these chromosomes, whenever they divide at the time of metaphase stage and anaphase stage, rather at anaphase stage they will start dividing, means getting separated from each other. You understand what I said? This chromatid will get separated from this chromatid and they will get separated. Here this is a point where spindle apparatus will be pulling it. This is a point where spindle apparatus will be pulling it. So they are pulled away from each other. And whenever they are pulled away from each other during anaphase stage of cell division, they will acquire some shapes. So metacentric chromosome will acquire V-shaped. Okay, why V-shaped? Reason is very simple. Both the arms are equal. And that's how whenever it is picked up and the chromosome is running after spindle fiber, it will appear V-shaped. Okay. Then submetacentric chromosome will be appearing L-shaped. Why L-shaped? Because see here, एल हम लोग ऐसे बनाते हैं, तो एक जो है इसका आर्म छोटा रहेगा, दूसरा जो आर्म है बड़ा रहेगा. सो वेन एवर इट इज पुल्ड, इट इज पिक्ड अप ओवर हियर, स्पिंडल फाइबर इसको यहाँ पे पिकअप करेगा और खींचेगा. खींचेगा तो क्या होगा, एक आर्म छोटा दिखेगा, एक आर्म बड़ा दिखेगा, तो इट विल डेवलप shape like L. So it is called as L-shaped chromosome. Then third is acrocentric chromosome. One of the arms is very short, other is very long. So when this is pulled, it will get a shape like J, English letter J. J का जैसा दिखेगा, and that's why it is called as J-shaped chromosome. And lastly, there is only one arm available, other arm is absent, so it will be called as I-shaped chromosome. ठीक है, English letter I जैसे दिखेगा. So metacentric chromosome is V-shaped, submetacentric chromosome is L-shaped, acrocentric chromosome is J-shaped, and lastly telocentric chromosome is I-shaped. Okay, अभी MCQ में क्वेश्चन आएगा L-shaped chromosome is dash, तो अभी आपको आएगा आंसर उसका, L-shaped chromosome will be submetacentric chromosome. But whenever we are discussing many questions at that particular time, you might not be able to get it. So just apply logic about their arms and you can get the basic feature. ठीक है, so this is we can say. So first हम लोगों ने देखा कि chromosomes are categorized on the basis of their shape, rather size we can say, बाद में position of centromere. Then one more category we can add over here, that is P:Q arm ratio. क्रोमोसोम के ऊपर सेंट्रोमियर है, बिकॉज ऑफ सेंट्रोमियर व्हाट हैपेंस, there are two arms made, both these arms are given different names, P arm and Q arm. So depending upon their length we can take a ratio. So what I said, P:Q ratio, यहाँ पे P:Q ratio एक आएगा, 1 आएगा. Here P:Q ratio will be less than 1, here it will be still less than 1, and here it will be 0. ठीक है, so remember this idea. So depending upon P:Q ratio we can categorize chromosome. अभी ऐसे Q that we have to understand. So autosomes and sex chromosomes, if we take into consideration, we have to have some ideas. First we can say is autosomes are non, क्या कहा है, autosomes क्या है, are homologous chromosomes, and they form pairs. Sex chromosomes are partially homologous, and they form pairs, but they are partially homologous. Sex chromosomes are not completely homologous. Then second we have to understand is sex chromosomes involved in formation of sex or determination of sex, whereas autosomes are not involved in determination of sex. That is a comparison we are making now between sex chromosomes and autosomes. This is one more category where we can distinguish or divide the chromosomes into certain groups like sex chromosomes and non-sex chromosomes. In case of sex chromosome, Y chromosome is shorter, and in autosomal pairs both the chromosomes are of equal length. तो ये एक और difference है. That's why sex chromosomes are also called as heterosomes. Hetero का मतलब होता है different, they are having different looking. In case of sex chromosome pair, the position of centromere may not be the same. सीधी बात है, एक क्रोमोसोम छोटा है, एक क्रोमोसोम बड़ा है, तो position of centromere is not same. And in case of autosomal chromosome pair, the position of centromere is exactly same. So this is the basic difference between the two chromosome types. Remember it very well. Now we have to understand the human karyotype. Karyotype क्या होता है? Karyotype is actually called as ideograph or ideogram. It is study of chromosomes. We have to take chromosomes and categorize them, or grouping of chromosomes will be called as ideograph or karyotyping. कैसे करते हैं पहले उसको देख लेते हैं. There is a methodology. ठीक है, so you have to take few cells, few dividing cells rather. As far as those dividing cells are concerned, you have to make a culture of these cells. Then few cells you have to mount on slide, color them, then stain the slide so that the cells will accept the stain, chromosomes will get the stain. तो chromosomal stain के साथ हम लोग उसको stain कर सकते हैं. Remember acetocarmine नाम का एक stain होता है. I repeat, acetocarmine is a stain which will be used for coloring or staining the chromosomes. तो acetocarmine को use करेंगे तो chromosomes का coloring हो जाएगा. And then what we have to do, we have to take a photograph of this nucleus, magnify the photograph. From this photograph you have to take the chromosomes away, मतलब क्या, हर एक क्रोमोसोम को काट काट के अलग करने का. And then you have to arrange the chromosomes in their proper order depending upon, again now we have to say first is size of chromosome. तो बड़ा क्रोमोसोम पहले लगेगा, उससे छोटा बाद में लगेगा, उससे छोटा बाद में लगेगा, इस तरह से from descending order of the size we have to arrange the chromosomes. Second criteria I told you, the position of centromere. Depending upon the position of centromere, chromosomes are of 4 types, so accordingly we can arrange them. Then we have to keep autosomes separate and heterosomes separate, means sex chromosomes separate. तो ऑटोसोम्स का ग्रुप पहले आएगा और सेक्स क्रोमोसोम्स का ग्रुप बाद में आएगा. थर्ड क्राइटेरिया हो गया. फोर्थ क्राइटेरिया इज P:Q आर्म रेशियो. P:Q आर्म रेशियो, इसको हम लोग कहेंगे एक और क्राइटेरिया. सो डिपेंडिंग अपॉन दिस क्राइटेरिया ऑल्सो वी कैन डिस्टिंग्विश द क्रोमोसोम्स और वी कैन सेपरेट द क्रोमोसोम्स फ्रॉम ईच अदर. ओके, मतलब कैटेगराइज करने में उसको मदद हो जाएगा. एंड वन मोर थिंग इज देयर, दिस इज कॉल्ड एज बैंडिंग पैटर्न. थोड़ा सा इसके बारे में डिस्कस कर लेते हैं. व्हाट इज बैंडिंग पैटर्न? देखो ये सब अपने बुक में नहीं दिया हुआ है, बट टेकिंग इनटू कंसिडरेशन वी आर ऑल गोइंग फॉर नीट, आई एम एक्सप्लेनिंग इट टू यू. ठीक है, जितना बुक में है उतना मैंने स्लाइड में इंक्लूड कर दिया है, पावर पॉइंट प्रेजेंटेशन में इंक्लूड कर दिया है. एक्स्ट्रा पार्ट आई एम डूइंग इट ओरली. गॉट इट वेरी वेल. सो व्हाट इज बैंडिंग पैटर्न? If you take a particular stain and stain the chromosome, it will get stained in different fashion. You know very well, heterochromatin and euchromatin parts are there. I told you heterochromatin accepts dark color and euchromatin accepts light color. So it develops a banding pattern. मतलब अपने को पता नहीं ना क्रोमोसोम के ऊपर हेटेरोक्रोमेटिन वाला पार्ट कहाँ पे है और यूक्रोमेटिन वाला पार्ट कहाँ पे है, अपने को नहीं पता. सो एक क्रोमोसोम को स्टेन करने के बाद एक बैंडिंग पैटर्न अपने को मिल जाएगा. इफ यू मेक यूज ऑफ अ सेम स्टेन एंड यूज chromosomes for staining it, in that case if it is possible that 2 or more chromosomes have same bending pattern, then they are of same type. मैंने 2 और more कहा, लेकिन more नहीं होगा, सिर्फ 2 chromosomes होंगे, that will show same bending pattern. And such kind of chromosomes, be sure they are called as homologous chromosomes. ठीक है, तो homologous chromosomes कैसे होते हैं? They are having same size, they are having same position, right, of centromere. Centromere का position same रहेगा, same P:Q ratio and same bending pattern. सेक्स क्रोमोसोम तो अलग ही रखते हैं हम लोग, उसको उसके मामले में नहीं लाएंगे अभी. सो होमोलोगस क्रोमोसोम के लिए क्या बनता है? सेम साइज, same position of centromere, same binding pattern and same P:Q ratio. Now depending upon all these things which we discussed, तो मैंने कुछ क्राइटेरिया आपको बताए, क्रोमोसोम्स को कैटेगराइज करने के लिए क्राइटेरिया बताए. पहला बताया था साइज, देन पोजिशन ऑफ सेंट्रोमियर, देन P:Q रेशियो, देन बाइंडिंग पैटर्न एंड ऑटोसोम्स और सेक्स क्रोमोसोम्स. ऑटोसोम सेक्स क्रोमोसोम अभी हम लोग एंड में रख रहे हैं. सो डिपेंडिंग अपॉन दिस 5 क्राइटेरिया we can categorize the chromosomes. Chromosomes के हम लोग ग्रुप्स बना सकते हैं एंड उस ग्रुप्स में क्रोमोसोम्स को हम लोग अरेंज कर सकते हैं. Whenever we arrange the chromosomes in a different groups like this, that display will be called as karyotype or ideograph. Fine. This technique of ideograph or karyotype was discovered so as to understand the faulty chromosomes. कभी कभी अपने बॉडी में फॉल्टी क्रोमोसोम्स होते हैं या नंबर वाइज ऑल्सो दे आर मोर और लेस. टू अंडरस्टैंड वेदर द क्रोमोसोम्स आर नॉर्मल मीन्स यूप्लॉइडी और एबनॉर्मल एन्यूप्लॉइडी. सी आई हैव मेड यूज ऑफ 2 वर्ड्स. यूप्लॉइडी मैंने आपको पहले बताया था, नॉर्मल सेट ऑफ क्रोमोसोम्स को हम लोग कहते हैं यूप्लॉइडी. ठीक है, कभी कभी क्या होगा, there will be something wrong with the chromosomes, so number of chromosomes will change. At that particular time we will call it as aneuploidy. Euploidy means normal set of chromosomes, normal number of chromosomes. But whenever we discuss aneuploidy, it indicates abnormal number or abnormal set of chromosomes. ठीक है, so try to understand these two things. उसको करने के लिए there are some chromosomal disorders like you must be knowing Klinefelter syndrome, Turner syndrome, Down syndrome, uh, trisomy of 18th chromosome, trisomy of 19th chromosome. ये सब disorders है. उस disorders को understand करने के लिए, and of course they are hereditary, and so to understand them properly well, this particular ideograph technique was discovered. Okay, fine. So let us go for it. We are having 7 groups of chromosomes. अपने बॉडी के अंदर 46 chromosomes है और 23 pairs of chromosomes है. These 23 pairs of chromosomes can be categorized into 7 groups: group A, group B, group C, group D, group E, F and G. Fine. So group A will have chromosome number 1 to 3 pair. Now remember, it is written chromosome 1 to 3 लिखा हुआ है, लेकिन इसको pair मानना है. So chromosome number pair 1, pair 2, pair 3 come over here. How these chromosomes are, just try to follow this. These are metacentric chromosomes. They are large and easily distinguished from the others because of its size and location of the centromere. ठीक है, सो बड़े क्रोमोसोम्स है, मेटासेंट्रिक क्रोमोसोम्स है. बी ग्रुप कंसिस्ट ऑफ क्रोमोसोम नंबर 4 and 5 ओनली. दो ही क्रोमोसोम के पेयर्स है. सो इट हैज गॉट 2 लार्ज साइज क्रोमोसोम्स एंड दे आर सब मेटासेंट्रिक. तो पहले और दूसरे ग्रुप में डिफरेंस क्या हुआ? फर्स्ट ग्रुप इज मेटासेंट्रिक, सेकंड ग्रुप इज सब मेटासेंट्रिक. ग्रुप सी, क्रोमोसोम नंबर 6 to 12 means, see how many pairs are there. 7 pairs of chromosomes will be there, and of course X chromosome. X chromosome is also of this particular category. Sex chromosome में दो chromosome होते हैं ना, X chromosome and Y chromosome. So X chromosome is kept in third group. Okay, so chromosome number 6, 7, 8, 9, 10, 11, 12, 7 pairs हो गए. मैंने गिना के बताया है, पहले मैंने ऐसे ही बताया था. These chromosomes are metacentric type of chromosomes and medium sized. Remember, okay, and submetacentric type. Okay, metacentric chromosomes भी होते हैं इसके अंदर, submetacentric chromosomes भी होते हैं, but they are all medium size, and that's why they are kept over here in the third group. Group number 4, D, chromosome number 13, 14, 15 pair will be acrocentric type of chromosomes of medium size and has got satellite chromosome. That's why they're kept separately. So if a question is asked which chromosomes have got satellite, then it is 13, 14, 15. 3 pairs of chromosomes are having satellite. Then group F, chromosome number 16, 17, 18, 3 chromosomes again. These are metacentric type of chromosomes and small sized with metacentric. Group F consists of chromosome number 19 and 20, 2 pairs. So very small and metacentric type of chromosomes. So they are available over here. Group G is chromosome number 21 and 22 and Y chromosome. This will be consisting of acrocentric chromosomes. They are very small and have got satellites except Y chromosome. So satellites are found in 5 chromosomes. Chromosome number 13, 14, 15, 21, 22. याद रखना बराबर से. तो ये एमसीक्यूज में पूछा जाएगा. वाई क्रोमोसोम एक्रोसेंट्रिक होने के बावजूद उसके ऊपर सैटेलाइट नहीं है. ठीक है, सो रिमेंबर दिस वेरी वेल. सो ग्रुप ए कंसिस्ट ऑफ 3 pairs, ग्रुप बी कंसिस्ट ऑफ 2 pairs, ग्रुप सी कंसिस्ट ऑफ 7.5 pairs. Now how this 7.5 pairs, 6 to 12 is complete pair and 1 X chromosome. Group D consists of 3 pairs, 13, 14, 15. Group E consists of 16, 17, 18, 3 pairs. Group F and Group G consist of 2 pairs of chromosomes each. ठीक है, तो 22 pairs तक यहाँ पे दिया हुआ है. X chromosome over here and Y chromosome over here will become a pair of sex chromosome that also is to be considered, but they are hetero, है ना, heterosomes, अलग अलग होते हैं दिखने के लिए, and even are placed at different place or different groups. So X chromosome belongs to C group and Y chromosome belongs to G group. They are different in their positions also. So that's how the ideograph is considered or taken into consideration. So 46 chromosomes can be divided into this particular 7 different groups. Okay, अभी ये ideograph कैसे दिखता है, that we need to understand. So ये अपना human ideograph है. Now have you observed one thing over here in ideograph? We have taken into consideration male for giving ideograph, because male ideograph is complete, female ideograph is not complete है. Female ideograph is not complete. क्या मतलब है? Females don't have got Y chromosome, males have got Y chromosome, females don't have got Y chromosome. तो पूरे chromosomes female में include नहीं होते हैं, and that's why female ideograph is said to be incomplete. Male ideograph is said to be complete because it consists of both X chromosome as well as Y chromosome. ठीक है, X chromosome as well as Y chromosome both are present, and that's why it is called as complete ideograph. So this is one picture of ideograph. You could see group number 1 means A group, B group, C group is the biggest one. ठीक है, then D group, E group, F and G group. X chromosome belongs to third group and Y chromosome belongs to seventh group. Fine. So mind it very well. This is how we have to consider ideograph. Then we have to understand comparison between X and Y chromosomes. Okay, these are called as sex chromosomes, or we can call it as heterosomes, or we can call it as gonosomes. Sex chromosomes because they determine sex or help in determining sex. That's why sex chromosomes. Gonosomes, again same thing, related to sex only. So involved in determination of sex. That's why gonosomes. And third, I told you they are called as heterosomes because they look different from each other. That's why they're called as heterosomes. So let us make a comparison between X chromosome and Y chromosome. They are looking different from each other. How and what are the features? So on one side we have got X chromosome, other side we have got Y chromosome. X chromosome is having a shape like that of English letter X. That's why it is called as X chromosome. Y chromosome is having shape like that of Y, English letter Y. That's why it is Y chromosome. Then we have to consider second point. X chromosome is linear and metacentric in nature. Linear का मतलब क्या हुआ? Straight line जैसा रहेगा. But Y chromosome is having a bent arm and it is acrocentric in nature. ठीक है? Sometimes it is said that it is acrocentric in nature. Why bent arm? It is having a short. Now see, acrocentric का मतलब क्या होता है? The centromere is towards the end but not exactly at the end. So this makes one arm, that is P arm, shorter and Q arm longer. Sorry. So when chromosome is kept like this, it will not remain linear. It will remain like this, bent. It will have a bent arm. तो एक ऊपर का छोटा सा पार्ट उसका bent रहेगा. That's what is called as bent arm is available and acrocentric in nature. X chromosome is longer than Y, whereas Y chromosome is shorter than X. Okay, taking into consideration bent arm also. X chromosome is found in both sexes, even males as well as females. Okay? In case of males, it is available in single dose. In case of females, it is available in double dose. Hope you must have understood single dose, double dose. It is 1 in males and 2 in females. Okay, but Y chromosome is purely found in males only. That's what is also called as Hollandric chromosome, wholly masculine. Okay, masculine का मतलब male वाला chromosome and feminine means female वाला chromosome. So X chromosome is responsible for producing or generating feminine characters. Y chromosome is responsible for producing masculine characters found in males only, and that's why called as Hollandric chromosome. I repeat this. Okay. Then Y chromosome, X chromosome, if you take into consideration, X chromosome is longer than Y chromosome, we have said, and Y chromosome is shorter than X. So this extra length of X chromosome is considered as non-homologous part to Y chromosome, whereas in case of Y chromosome, there is a bent arm. This bent arm is said to be non-homologous to X chromosome. Now remember, this X chromosome and Y chromosome, they are having partly homologous nature and partly non-homologous nature. मतलब X chromosome का extra length और Y chromosome का bent arm non-homologous part है. बाकी के जो part है उसमें से ये homologous nature दिखाते हैं. Do not show SRY, sex determining region जो है इसके ऊपर नहीं रहेगा. It is present on Y chromosome only and so shows SRY. Okay, So this is how we can say a comparison between the two chromosomes, X chromosome and Y chromosome. This can be asked as a 2-mark question. Distinguish between X chromosome and Y chromosome. You are supposed to write the appropriate points. Appropriate points का मतलब समझ में आया? If your writing has shape like X alphabet, and against that if you write bent and acrocentric, आप लोग बोलोगे सर दोनों पॉइंट बराबर लिखे मैंने. Yes, दोनों पॉइंट बराबर है. I even agree, but they are not points to compare because this point and this point are totally different from each other. So though there are correct points told or written by you, you will not get marks for that. Okay, so similar comparison points are necessary. तो ये हमेशा याद रखें, whenever you are comparing, you have to give similar comparing points for both the criteria. Fine. Okay, let us move ahead for understanding more about X and Y chromosomes. Yes, this is the picture which will depict or indicate that what is the difference between them. You could see this is metacentric chromosome, this is acrocentric chromosome. X chromosome is larger or longer than Y, Y is shorter than X. Now see, this short arm will act as a bent arm. Okay, it is said as pseudo-autosomal region, and sex limited region is present on it. It is this, and pseudo-autosomal region, long arm. Okay, so remember this very well. Length of this chromosome is given over here. So much base pairs. Now you might read about it. Okay, gene number is around 160 and gene number on Y chromosome is 1228. So ये थोड़ा सा याद रखने की कोशिश करो. Here it is, X chromosome, inactivation center, many genes are present over it. Now ये जो extra length उसकी रहेगी, that is non-homologous part to Y chromosome. Fine. So that was a beginning of the chapter or mid of the chapter where we compared the chromosomes. Now we have to understand very important thing of this particular chapter, that is genetic linkage. In case of earlier half of this part, we understood Mendel's work. And at that particular time, we said that Mendel got complete dominance. And Mendel gave a law that is called as law of independent assortment. I repeat, the law of independent assortment was given by Mendel. What was the law saying? The law said chromosomes have got genes present on them, and genes for one character or genes for one trait do not depend on the genes for other character or trait to express. Law of independent assortment. And that's how we got mixed characters. Try to remember that yellow round we crossed with that of green wrinkled, and when yellow round was crossed with green wrinkled, we got 9 yellow round, then we got 3 yellow wrinkled, then we got 3 green round, and we got 1 green wrinkled. आप लोग बोलोगे सर वापस पीछे क्यों जा रहे हो? I'm just trying to make you understand that was independent assortment. मतलब क्या था? The two genes will not be dependent onto each other for their expression. हर एक जीन अपना अपना देख लेगा. तू मैं, तेरा तू देख, मेरा मैं देखता हूँ. But here, a work which was done by T.H. Morgan indicates that there is a linkage phenomena observed. मैंने कहा था कि मेंडल जी के बाद बहुत सारे साइंटिस्ट ने काम करना शुरू कर दिया जेनेटिक्स के फील्ड में, और जब ये लोग काम कर रहे थे तो उनको कुछ रिजल्ट्स मिले. Those results were not tallying with Mendel's results. Initially they thought that they were wrong. शुरुआत में उनको लगा कि अरे हम लोगों ने कुछ गलत काम कर दिया, मेंडल जी ने इतना अच्छा काम कर दिया है और हम लोग को वो रिजल्ट नहीं मिल रहे, तो हम लोग गलत है शायद से. But later on, when the experiments were repeated again and again and again and again, एक ही तरीका है ना अपने को सच और झूठ जानने का, एक ही तरीका है. जो जेनेटिक के एक्सपेरिमेंट्स है उसको बार बार करते जाओ, अगर वही रिजल्ट आपको मिलता है तो योर एक्सपेरिमेंट इज करेक्ट एंड दैट इज अ रूल. तो जब ये लोगों ने, पोस्ट मेंडेलियन लोगों ने एक्सपेरिमेंट्स करना शुरू कर दिया, उनके रिजल्ट्स टैली नहीं हो रहे थे, उन्होंने रिपीट किए एंड दे फाउंड दैट इट इज समथिंग डिफरेंट फ्रॉम मेंडल्स लॉज. एंड सो टी.एच. मॉर्गन सेड दिस एज लिंकेज. अभी लिंकेज का मतलब क्या होता है? लिंक करना मतलब आपको पता है, दो चीजों को लिंक करना मतलब दो चीजों को कनेक्ट करना. So this phenomenon is exactly opposite to that of independent assortment. ठीक है, इंडिपेंडेंट असॉर्टमेंट क्यों होता है उसका एक्सप्लेनेशन मेंडल जी ने दिया था. क्या एक्सप्लेनेशन था? He says, Mendel says, why the genes of one trait do not depend on other trait for expression. तो उन्होंने दो पॉसिबिलिटीज कहे थे. नंबर वन पॉसिबिलिटी इज either gene for one trait are present on one chromosome and gene for other trait is present on other chromosome. They are not related at all. दोनों जीन्स एक ही क्रोमोसोम के ऊपर नहीं रहेंगे. मतलब gene for color and gene for shape are differently present on different chromosomes and that's why they got independently expressed. दूसरी पॉसिबिलिटी उन्होंने यहाँ पे कही थी कि दे माइट बी प्रेजेंट ऑन सेम क्रोमोसोम बट एट अ वेरी लॉन्ग डिस्टेंस. मतलब जीन फॉर कलर एक एंड पे है और जीन फॉर शेप दूसरे एंड पे है एंड दैट्स व्हाई देयर इज नो कनेक्शन बिटवीन देम. बट टीएच मॉर्गन व्हेन स्टडीड something, he found that a gene for one trait depends on gene for other trait for its expression and that's how we called it as linkage. This phenomenon was proved by using another individual for genetic study that is commonly used nowadays, Drosophila melanogaster. Okay, commonly we call it as fruit fly. मेंडल जी ने क्या यूज किया था? Pisum sativum plants were used by Mendel, but this person T.H. Morgan he made use of another organism. This is animal, Drosophila melanogaster fruit fly. अभी आपने देखा रहेगा fruit fly कि नहीं देखा रहेगा मुझे नहीं पता, but let me make it very clear. देखना है fruit fly तो एक common चीज है. आपके घर में कभी फल लाके रख देते हैं, खास करके बनाना, चिकू, है ना. ये फल ऐसे है कि उनका जो स्मेल है ना वो स्प्रेड हो जाता है पकने के बाद, बहुत ज्यादा अमाउंट में स्प्रेड हो जाता है and that attracts some very small flies. Those flies which are attracted by the smell of fruit are called as fruit flies. Scientifically Drosophila melanogaster उसका नाम है. फाइन. अभी आपको उसको देखना है तो बहुत सिंपल तरीका है, आसान तरीका है. यू कैन वॉच देम. आई विल टेल यू वन स्मॉल थिंग व्हाट यू विल परफॉर्म. टेक ए स्मॉल कंटेनर, कटोरी वगैरह अपने घर में होते हैं, छोटी सी कटोरी ले लो. ओके. देन या डिश ले लो छोटी सी. उस डिश के अंदर एक बहुत पका हुआ बनाना जो है ना, उस बनाना को क्रश करके उस डिश के अंदर रख दो. देन एक बड़ा सा ग्लास का जार ले लो. ठीक है, उस जार के अंदर इसको रख दो. जो क्रश किया हुआ बनाना है उसके अंदर उसको रख दो, जार के अंदर टोकरी में आपने रखा हुआ है. कुछ देर उसको ऐसे ही छोड़ दो घर में, शांत जगह पे, मतलब जहाँ पे कोई आदमी बहुत बार आता जाता नहीं रहेगा. एक कोने में रख दो उसको जहाँ पे ठंडक हो. You will observe that after some time inside the jar there are some small flies are gathering. Now cork the bottle. कांच का बॉटल जो लिया था, जार लिया था आपने, उसको कॉर्क कर दो, बंद कर दो. द फ्लाइज आर ट्रैप्ड इनसाइड. नाउ यू कैन ऑब्जर्व देम. ठीक है, ऑब्जर्व क्यों करना है? बिकॉज वी हैव टू अंडरस्टैंड देयर मॉर्फोलॉजी. जो एक्सपेरिमेंट मेंडल जी, जो एक्सपेरिमेंट टीएच मॉर्गन जी ने यहाँ पे किया है, उस एक्सपेरिमेंट को अगर अपने को समझना है तो ये थोड़ा सा आपको उसके बारे में मालूम होना चाहिए. ओके. सो जैसे मैंने पहले एक्सप्लेन किया था, ये जो लिंकेज वाला फिनोमिनन है, इट इज एग्जैक्टली डिफरेंट और इट इज एक्सेप्शनल टू मेंडल्स लॉ ऑफ इंडिपेंडेंट असॉर्टमेंट. तो मैंने दोनों का डिफरेंस कहा है. मेंडल्स लॉ ऑफ इंडिपेंडेंट असॉर्टमेंट में क्या कहा था कि जीन्स फॉर वन ट्रेट डू नॉट डिपेंड ऑन जीन्स फॉर अदर ट्रेट फॉर देयर एक्सप्रेशन. बट ये टीएच मॉर्गन जी कहते हैं genes for one trait do depend on gene for others for their expression, means they always go hand in hand. Okay. अभी क्या लॉजिक है? मेंडल जी का लॉजिक क्या था? either the two genes of two different traits are present on two different chromosomes. ऐसे केस है तो forget it, भूल जाओ उसको. But take into consideration second condition. What Mendel had predicted, the two genes of two different two genes for two different traits, characters might be present on same chromosome and at two far ends. दो अलग अलग एंड पे रहेंगे. ओके. तो अभी ये जो स्टेटमेंट यहाँ पे मैंने दिया है उसको पढ़ो बराबर से. मैं पढ़ूंगा उसको, लिटरली पढ़ूंगा, मैं उसको बताऊंगा नहीं आपको. इफ two genes are close enough on one chromosome, then chance of crossing over, that is exchanging genetic fragments in between these two genes, is less. ओके, आगे क्या कहते हैं, frequency of these two genes being together is more and chance of recombining or recombinations is less. This is called as tight linkage or complete linkage or linkage. Linkage का मतलब क्या? The two genes are linked together, connected to each other. Okay, now let us discuss. ये तो अभी हम लोगों ने पढ़ लिया, अभी डिस्कस करना है. Now take into consideration one thread with you. 10 सेंटीमीटर का एक धागा आप लोगों ने ले लिया, ठीक है. आपको क्या करना है, 10 सेंटीमीटर का धागा आपने लिया है, एक पॉइंट के ऊपर उसको एक कलर का डॉट लगाना है और इमीडिएट दूसरे पॉइंट के ऊपर उसका दूसरा कलर का डॉट लगाना है 1 सेंटीमीटर के गैप में, और एक तीसरे कलर का डॉट आपको लगाना है थोड़ा सा दूर 4 सेंटीमीटर के गैप में. मतलब आपने 3 डॉट लगा दिए. पहले डॉट से लेके दूसरे डॉट का डिस्टेंस 1 सेंटीमीटर है, दूसरे डॉट से तीसरे डॉट का डिस्टेंस 4 सेंटीमीटर है, और पहले डॉट से तीसरे डॉट का डिस्टेंस 5 सेंटीमीटर है. होप यू मस्ट हैव अंडरस्टूड. आर यू अंडरस्टैंडिंग व्हाट आई एम ट्राइंग टू से नाउ? द डिस्टेंस बिटवीन द टू जीन्स इज इंपॉर्टेंट ओवर हियर. ये मायने रखता है. आप लोग बोलोगे उसका क्या संबंध है? ओके, लेट अस गो बैक टू meiosis. During meiosis, you must be knowing, during prophase 1, the sister chromatids remain together, but homologous chromosome come together. They form pairs. Then there is coiling of sister chromatids of opposite homologous chromosomes. And then there is exchange of genetic fragments. Hope you must have understood what I am talking about. There is chiasmata formation and crossing over. It is a best phenomenon that is done during the process of meiosis. Meiosis is done during gamete formation. So during gamete formation, the chromosomal fragments of homologous chromosomes are exchanged. मतलब वहाँ पे chromosome break होता है और reunite होता है. But what will happen? The chromosomes will break and make in a different fashion. That is what is called as crossing over. Here is the diagram. Okay, see, these are the two homologous chromosomes. These two are sister chromatids of one, the one which are in orange, and the brown one are the sister chromatids of the other chromosome. See what is happening. During the process of prophase 1, they are coming together. Chromosomes, homologous chromosomes are coming together. They twine around each other, coil around each other, and the points of contact are called as chiasmata. Okay, and at this particular point, the chromosomal fragments break and they are transferred to opposite pair of chromosome, that is homologous chromosome, and so the chromosomes will be grafted. After the chiasmata formation and crossing over, see what is happening. This sister chromatid is original of its type. This sister chromatid is original of its type, but the inner sister chromatids, they have got grafting of their homologous chromosome. Okay, so this process takes place between the two homologous chromosomes. The genetic material is exchanged. यहाँ पे एक ही जगह दिखाया है, ऐसे बहुत जगह पे ये हो सकता है. At many points, this process of chiasmata formation and crossing over takes place. Okay, this will take place between the non-sister chromatids of homologous chromosomes. Now try to understand this. I told you these two orange are sister chromatids, these two brown are sister chromatids. One entire brown and one entire orange are homologous chromosomes. So one orange and one brown are non-sister chromatids. उनके बीच में ये प्रोसेस हो रहा है. So what is said over here? Read this properly again. Genetic material is exchanged between non-sister chromatids of homologous chromosomes. Homologous chromosomes के बीच में होगा, लेकिन उनके non-sister chromatids के बीच में होगा. It will give new genetic combinations. यहाँ के जो जीन्स थे यहाँ पे चले गए, यहाँ के जीन्स यहाँ पे चले आए. Means it is giving new combinations, and this combination is nothing but the base of variation. नए कॉम्बिनेशन बनेंगे तो नए वेरिएशन भी बनेंगे. दैट्स व्हाट इज इंपॉर्टेंट. तो कमिंग बैक टू आवर थ्रेड विच आई हैड टोल्ड यू, 10 सेंटीमीटर का थ्रेड लिया आपने, एक डॉट लगा दिया 1 सेंटीमीटर के ऊपर दूसरे कलर का डॉट, और 5 सेंटीमीटर 4 सेंटीमीटर के ऊपर तीसरा डॉट. क्या लगता है आपको, इफ यू टेक दिस थ्रेड, hold it like this and try to break it randomly, will it be a chance that the thread will get broken between first dot and third dot more, or chance of getting thread broken between first and second dot broken more? जो डॉट पास पास में है उनके बीच में थ्रेड टूटने के चांसेस ज्यादा है कि जो दूर है उसके बीच में टूटने के चांसेस ज्यादा है? आंसर इज वेरी ऑब्वियस, यू मस्ट हैव थॉट द सेम. जो डॉट्स दूर दूर है उसके बीच में टूटने के चांसेस ज्यादा है. मतलब chiasmata formation and crossing over process is possible if the two genes are apart from each other on the same chromosome or homologous chromosome. ठीक है, homologous chromosome के ऊपर अगर ये अलग अलग जगह पे है तो ये प्रोसेस फास्ट होगा. बट इफ द टू genes are very close enough, then chances of breaking the chromosome between them and separating is less. Means chance of crossing over and chiasmata formation is less. Okay, so देखते हैं क्या होता है. This organism was used by T.H. Morgan, fruit fly, Drosophila melanogaster. Now try to observe the morphology of these two organisms. He used different organisms, one with brown body, another with yellow body, white eye and red eye. ये सही में होता है. मैंने कहा जो कि वो ऑर्गेनिजम्स को पकड़ के आपको देखना है तो आप लोग देख सकते हो. रियली इट इज पॉसिबल. ओके, एंड इट विल बी सीन. टीएच मॉर्गन कंसिडर्ड फॉलोइंग ट्रेट्स. अभी उन्होंने एक्सपेरिमेंट किया है इसके ऊपर, तो कौन से ट्रेट लिए? ही सिलेक्टेड फर्स्ट ट्रेट, दैट इज बॉडी कलर. बॉडी कलर इज अवेलेबल इन टू फैशन, ब्राउन बॉडी एंड येलो बॉडी. ओके, अभी इन्होंने अपने एक्सपेरिमेंट में जीन्स को दिखाने के लिए थोड़ा सा अलग तरीका यूज किया. मेंडल जी ने क्या यूज किया था? कैपिटल लेटर फॉर डोमिनेंट जीन एंड स्मॉल लेटर फॉर सेम स्मॉल लेटर फॉर रिसेसिव जीन. बट इन्होंने थोड़ा सा अलग तरीका यूज किया है. फॉर डोमिनेंट कलर फॉर डोमिनेंट ट्रेट व्हाट ही सेज, a small letter with plus sign, and for recessive trait only a small letter. ठीक है, तो brown body being dominant character will be indicated by y plus, and yellow body being recessive character will be indicated by small y. Similarly, second trait he took into consideration, color of eye. He found that it is red or white. Red is dominant over white, and so he took W plus as red eye and W as white eye. तो क्या हो रहा है समझ में आया? रेसेसिव कैरेक्टर के लिए वो स्मॉल लेटर यूज कर रहे हैं और डोमिनेंट कैरेक्टर दिखाने के लिए उस स्मॉल लेटर के ऊपर प्लस साइन डाल रहे हैं. थर्ड कैरेक्टर ही कंसिडर्ड वाज wing size. Normal wing means the wing which is growing to normal size. He said it is M plus, and miniature wing as W. So sorry, it has to be M, small m. Fine. So M plus and M wing size उन्होंने रख लिया. One more thing is important over here. In case of Drosophila melanogaster, male and female sexes are different. Male consists of X and Y both chromosomes, and females have only X chromosomes. All the three characters, or all the three genes of three different characters which we took, कौन से ट्रेट्स लिए हम लोगों ने? बॉडी कलर, कलर ऑफ आई एंड साइज ऑफ विंग. तो ऑल दीज जीन्स फॉर कलर बॉडी, बॉडी कलर, जीन फॉर कलर ऑफ आई एंड जीन फॉर साइज ऑफ विंग are all present on X chromosome only. They are not present on Y chromosome. So Y chromosome is literally playing no role in this. ठीक है? So no crossing over or its effect will be seen in the present generation individual or parent generation individual. क्यों ऐसा होगा? क्योंकि parents में crossing over नहीं होगा. Parents we are selecting as homologous type. Okay. Now let us go for the experiments. Morgan performed two experiments simultaneously. Okay, which two experiments were performed? Experiment number 1A was considered like this. He took wild male with brown body and red eye. मैंने कहा था wild का मतलब क्या है, लास्ट सेशन में कहा है wild genes are dominant genes. Okay, so he took male wild, matlab kya? The body color is brown and color of eye is red, and he crossed this with female. Female was mutated, having yellow body color and white eye. In the second experiment, he considered wild male with red eye and normal wing, whereas female was with white and miniature wing. What is so क्या common है दोनों में? Red eye and red eye is common over here. White eye and white eye means color of eye is common. This color of eye he is testing with other two traits, size of wing and body color. ठीक है, अलग अलग experiments में ये अलग अलग कर रहे हैं. So see what is happening, what he has taken. This is he took as a parent generation. So cross A was having this experimental cross, means wild male. Wild male means this, having yellow body color and red eye. Yellow body color will be indicated, sorry, brown body color will be indicated with the help of Y plus, and red eye will be indicated with the help of W plus. Female will be having yellow and white, means yellow body color and white eye. इस एक्सपेरिमेंट में क्या किया? He took W means white eyes and crossed them with that of miniature wing, and here he took male with red eyes and normal wings. दोनों एक्सपेरिमेंट्स को हम लोग साइमल्टेनियसली करते जाएंगे. Now these individuals, they will be undergoing a process of gamete formation. During gamete formation, what is necessary? Chiasmata formation and crossing over. क्या यहाँ पे क्रॉसिंग ओवर और चियास्माटा फॉर्मेशन होगा कि नहीं होगा, इधर ही देखना पड़ेगा. मेल में तो नहीं होगा बिकॉज दीज आर नॉन होमोलोगस क्रोमोसोम्स, है ना. एक्स क्रोमोसोम एंड वाई क्रोमोसोम आर नॉन होमोलोगस क्रोमोसोम्स, एंड दैट्स व्हाई देयर इज नो चियास्माटा फॉर्मेशन एंड क्रॉसिंग ओवर. बट इन केस ऑफ दिस देयर विल बी चियास्माटा फॉर्मेशन एंड क्रॉसिंग ओवर पॉसिबल, बट द जीन्स आर सेम, है ना. Y and W, here also it is Y and W, the genes are same. The genes being same, though ये जीन यहाँ पे गया, ये जीन यहाँ पे आया, कुछ भी उल्टा सुल्टा हुआ तो it will be unnoticed. Same thing is true over here. Genes for these two traits are same on X chromosomes, so there is no effect of chiasmata formation and crossing over in parents. That was a wording which was given earlier in the earlier slide. ठीक है, ये क्यों नहीं होगा? क्योंकि उनका they are पेरेंट्स कैसे है? प्योर ब्रीड्स है. प्योर ब्रीड्स का मतलब आपको पता है, दे आर कीपिंग देयर कैरेक्टर्स, हाँ, और दे आर होमोजाइगस डोमिनेंट और होमोजाइगस रिसेसिव है. सो देयर जीन्स आर सेम, सो देयर इज नो इफेक्ट ऑफ चियास्माटा फॉर्मेशन एंड क्रॉसिंग ओवर. आफ्टर परफॉर्म performing the experiment, he got result of F1 generation. क्या रिजल्ट मिला? तो कैसे किया उन्होंने एक्सपेरिमेंट? लेट अस टेक इनटू कंसिडरेशन. क्रॉस ए, parents are like this that we have showed, then they will be having F1 generation like this. Now what will happen? इनके गैमेट्स कैसे बनेंगे? Y and W, here will be Y plus W plus. यहाँ पे गैमेट्स कैसे बनेंगे? W and M, here it will be Y plus M plus. क्या होगा exactly? This X chromosome of female will get separated from this X chromosome. But दोनों X chromosome के ऊपर same genes है, so there is no difference. But here there is something different. This X chromosome will get separated from Y chromosome. So female is forming one type of gamete and male is forming two types of gametes. Getting this? So क्या होगा देखो, when this X chromosome comes with this X chromosome of female, you will get this combination, and when this X chromosome of female comes with this Y chromosome of male, it will give this combination. अभी देखो क्या होगा. This condition is Y plus, this condition is WW plus. तो heterozygous condition तैयार हुआ. But one gene being dominant, it will be expressing itself as per the Mendel's first law of dominance. Okay, and that's why this individual being having two X chromosomes is female and wild type. Wild type का मतलब क्या हुआ? Having brown body and red color of eyes. ये जीन सप्रेस रहेगा. स्मॉल y स्मॉल w जीन सप्रेस रहेंगे. यहाँ पे क्या होगा? The genes are present only on X chromosome. Similar genes are not present on Y chromosome. So as there is one X chromosome and one Y chromosome, this individual is male and both the recessive genes will express themselves. So it will be yellow and white. So color of body will be yellow, color of eyes will be white. ठीक है, यहाँ पे क्या होने वाला है? Similar case, but this female will become wild type, means having red eyes and normal wings. Okay, this male will be mutated type, having white color of eyes and miniature wings. Remember that in this case females are heterozygous. Females are heterozygous, means they are heterozygous brown and red. Here it is heterozygous red eyes and normal wings. Getting it clear? Very well. So as far as this concept is concerned, we have got male with yellow body color and white eye. Female will be having brown body color and red eye. In this particular second experiment, male with white eye and miniature wing, female with red eye and normal wing. Now he will go for selfing. अपना रूल क्या है? जेनेटिक एक्सपेरिमेंट का रूल क्या है? F1 जनरेशन को अपने को सेल्फिंग करवाना होता है. सो सेल्फिंग करेंगे. सेल्फिंग करेंगे मतलब क्या करेंगे? एक ही जनरेशन के या सिमिलर फिनोटाइप के या सिमिलर जीनोटाइप के ऑर्गेनिजम्स को अपने को ब्रीड करना होता है. सो F1 जनरेशन वाज लाइक दिस. अभी सेल्फिंग करने के लिए अपने को क्या करना पड़ेगा? उनके जीन्स को यूज करना पड़ेगा. सो क्या है यहाँ पे? मेल with yellow body and white eye is crossed with female with that of brown body and red eye. इस एक्सपेरिमेंट में क्या हुआ? दूसरे एक्सपेरिमेंट में क्या हुआ? मेल with white eye and miniature wing is crossed with that of female with red eye and normal wing. अभी क्या होगा? There will be crossing over in female. फीमेल के अंदर क्रॉसिंग ओवर हो सकता है, मेल में भी हो सकता है कि नहीं हो सकता है? मेल में नहीं होगा, कुछ फायदा ही नहीं है. X chromosome and Y chromosome are non-homologous chromosomes. So there will be crossing over in case of female, and there will be 4 different types of alternatives are generated. I told you last time, if an individual is heterozygous in 2 traits, what will happen? There will be 2:2 types of gametes produced. 2:2 मतलब 4 है ना. फीमेल देखो क्या है? फीमेल इज हेटेरोजाइगस इन 2 traits and that's why 2:2 means 2:2 means what? It is sorry 4 gametes are produced. ओके, तो फीमेल 4 गैमेट बनाने वाली है. मेल क्या करेगा? मेल के अंदर रिकॉम्बिनेशन नहीं होने वाला है, क्रॉसिंग ओवर का प्रोसेस नहीं होने वाला है. जस्ट X chromosome and Y chromosome will separate. Got it? Very well. So female is going to produce 4 different types of gametes. Male is going to produce 2 different type of gametes. So how many recombinations will be there? The total number of recombinations will be gametes produced by one individual into gametes produced by another individual. So 4 into 2, that will be 8 possibilities will be there. So we have to consider now the result of F2 generation, and that is formation of recombinations. Recombinations का मतलब क्या? कुछ तो अलग अलग तैयार होगा यहाँ पे. What is different? Now see what has happened very clearly. पहले हम लोग X क्रोमोसोम के बारे में यहाँ पे देख लेते हैं. चारों जगह पे वैसा ही होने वाला है. See, this is one X chromosome with Y plus W plus. This is another X chromosome यहाँ पे देखते हैं पहले, अनदर एक्स क्रोमोसोम विथ वाई प्लस एंड डब्ल्यू, यहाँ पे क्या हुआ वाई एंड डब्ल्यू, एंड यहाँ पे क्या हुआ वाई डब्ल्यू प्लस. तो हियर बोथ द जीन्स आर डोमिनेंट. यहाँ पे जीन फॉर कलर ऑफ बॉडी इज डोमिनेंट, कलर ऑफ आईज इज रेसेसिव. यहाँ पे जीन फॉर color of body as well as color of eyes both are recessive, and here gene for color of body is recessive and color of eyes is dominant. This is coming in contact with Y chromosome. तो चारों Y chromosome के साथ आ रहे हैं. अभी ये क्रोमोसोम और ये क्रोमोसोम सेम है, ये क्रोमोसोम और ये क्रोमोसोम सेम है, ये क्रोमोसोम और ये क्रोमोसोम सेम है, ये क्रोमोसोम और ये क्रोमोसोम सेम है. ऊपर वाले क्रोमोसोम्स फीमेल के हैं, नीचे वाले क्रोमोसोम्स मेल के हैं. नाउ दिस क्रोमोसोम ऑफ फीमेल इज कमिंग विथ क्रोमोसोम एक्स क्रोमोसोम ऑफ मेल. यहाँ पे y के साथ आया था, यहाँ पे x के साथ आएगा. सो सी व्हाट इज हैपनिंग. ओके, एंड सी द रिजल्ट. This will be having wild type, means brown body, red eye. This will be having white eyes and brown body. This will be having yellow body and white eyes. And this will be having yellow body and red eyes. Similarly here also it will be wild type, क्योंकि ये dominant genes खुद को express करेंगे. यहाँ पे क्या हो जाएगा, color of eyes will be white and color of body will be brown. यहाँ पे क्या होगा, color of eyes will be white and color of body will be yellow. यहाँ पे क्या होगा, color of eyes will be red and color of body will be brown. ठीक है, so remember this very well. Now see here, this is wild type of organism and this is another organism which is recessive. So अपने पास दो इंडिविजुअल्स है, एक वाइल्ड टाइप मतलब डोमिनेंट वाला है और एक रेसेसिव वाला है पूरा. तो these are showing, these two organisms are showing parental characters and these organisms are showing also parental characters, whereas these individuals are showing recombinations. What does it mean recombinations? उनमें characters mix हुए हैं, उनमें characters mix हुए हैं. So this is very important case. And now see at the top what is said. सबसे ऊपर क्या लिखा हुआ है, parental character types are 98.7% and recombination types are just 1.3%. तो ज्यादा क्या मिला यहाँ पे, parental characters ज्यादा मिले और recombinants कम मिले. जब मॉर्गन ने एक्सपेरिमेंट किया कलर ऑफ आईज और विंग साइज में सिमिलर एक्सपेरिमेंट किया हुआ है, देन सी द रिजल्ट. पैरेंटल कैरेक्टर्स वेर 62.8% एंड रिकॉम्बिनेशंस वेर 37.2%. ओके, अभी हम लोग इस रिजल्ट को कंपेयर करते हैं मेंडल जी के रिजल्ट के साथ. व्हेन मेंडल परफॉर्म डाइहाइब्रिड रेशियो, he got dihybrid ratio as 9:3:3:1. 9 individuals were yellow round, 1 individual was green wrinkled. So this 9 plus 10 is equal to 10 individuals were showing parental characters and 3 individuals which were yellow wrinkled and 3 individuals which were green round were showing recombinations. तो मेंडल के केस में क्या हुआ, आउट ऑफ 16 10 इंडिविजुअल्स आर शोइंग पैरेंटल कैरेक्टर्स एंड 6 इंडिविजुअल्स वे आर शोइंग रिकॉम्बिनेशंस. अगर इसको आप परसेंटेज में कन्वर्ट करोगे तो यू विल फाइंड दैट अराउंड 67% was parental characters in Mendel's laws or Mendel's experiment and 33 was recombinations. Try to look at this. यहाँ पे भी ऑलमोस्ट वही रिजल्ट मिला हुआ है. मॉर्गन्स एक्सपेरिमेंट 2 में अपने को मिला है पैरेंटल कैरेक्टर्स इन इंडिविजुअल्स 62.8% and recombinations are 37%. So what this experiment indicates? Experiment indicates these genes, means gene for color of body and color of eye, must be close enough. being close enough, they are being transmitted to next generation in a linked fashion, and that's how the frequency of these two being together is more. Frequency of these two being together is more, but frequency of recombinations is less. यहाँ पे क्या हुआ, frequency of two genes being together is less compared to this, and frequency of two genes forming recombinations is more. So in short, if recombinations formed are more, then it will be called as loose linkage, and if recombinations formed are less, it will be indicating tight linkage. Read what it is given over here. More organisms of parental type and less organisms with recombinations indicating tight linkages. Less organisms of parental type and more organisms of recombination type indicating loose linkage. मतलब क्या है, लिंकेज तो है but loose linkage or tight linkage. Those genes which are close to each other will be inherited in a linked fashion. Their frequency being together will be more. That's why it is called as tight linkage. And the others will not be there. And that's why it will be called as loose linkage. Now let us come to our statements once again. If two genes are close together, I'm concluding this now and concluding lecture also with these two statements. So try to listen this very carefully. So what I'm saying, on single chromosome, if genes for two different genes for two different traits are close enough, then chance of crossing over and what we can say, chiasmata formation is less. The chance of breaking of chromosome in between these two genes is less. So chance of recombinants is less, or frequency of recombinants is less, and frequency of parental characters is more. Such kind of case we will call it as tight linkage. Oppositely, if two genes of two different traits are away from each other on a single chromosome, chance of crossing over and chiasmata formation is more. Chance of breaking the chromosome between these two genes is more. So chance of recombinations is more, and chance of parental characters will be less, or frequency of recombinations is more. Frequency of parental characters is less, and it is called as loose linkage. So that is what the experiment was, and it showed exactly opposite result of that of Mendel's law of independent assortment. Okay, so go through the experiment once again in the book. I think I have explained it properly well. I have tried my level best to make you understand, but still once again, if you go through your book, it will be better. Okay, so what we'll do is we'll stop here.